Hi, this is Miss Litton. This is 7th period review, AP Biology, week before finals. Super excited. Say hi. Hi. Was that an under the breath cough I just heard? No. Okay. So yay, AP Unit 4. Shall we start by reviewing Chapter 13? Sure. Okay. Sure. All right. So that brings us to the lovely operon. What? Okay. Do you remember the parts of the operon? Yes. Okay. RNA polymerase binds to the promoter. Okay. Easy peasy lemon squeezy. Is the promoter part of the operon? Yes. What else is part of the operon?
The regulator.
Not the regulator. It's farther up. Operator. Yes. Who binds to the operator? Repressor. Who coded for the repressor gene? Farther upstream, not part of the operator, right? Yeah. Right. See the hash, right? Okay. And then there are, I saw people over there talking structural genes, yes? Yeah. That's part of it. Okay, so when you talk about the operon, you're really talking about what? Prokaryotic or eukaryotic cell? Eukaryotic. What? Prokaryotic. Prokaryotic. Operon is with a prokaryote. Operon is with a prokaryote. So anything that interferes with RNA polymerase's ability to bind, what are the two ways that you can interfere with RNA polymerase's ability to bind to RNA polymerase binds to the promoter? What? A repressor is binding to the operator, and that could get in the way. What's another way? No. Don't think negatively. What is it? That's all. So to the T, transcription factor. You hear, let's see if I can't non-keep teacher, right? Transcription factor, right? Remember, transcription factors, RNA polymerase that could need assistance to bind on, so a transcription factor would assist in that. You're thinking about cyclic AMP, right? And CAP, yes? And cyclic AMP goes up when glucose is down. Yeah. And so that is a positive control. Negative control is anything to do with repressor. Okay, and then we have two other words we need to think about. Inducible and repressible operon. Inducible and repressible. Hi, welcome. If it's inducible, am I currently transcribing those genes? No, but I can induce it. How can I induce something? If it's off for a reason, right? If it's off, that means the repressor is on the operator, right? And so RNA polymerase can't get on the promoter. How can I get the repressor away? What could I do? Yes? Okay, maybe not an enzyme, but something else that we talked about. Lactose is the example. And you call that an inducer. So it may not be an enzyme, okay? So you have an inducer. Usually you need an enzyme to break that, whatever that inducer is, down. So you would have an inducer like lactose, bind to the repressor, and pull it away because it cannot bind to both simultaneously. Now RNA polymerase gets on the promoter, transcribes those genes. Those genes, once translated, form enzymes that actually digest lactose. When all the lactose is digested, repressor is welcome to get right back on that operator. Make it logic out. Okay? I don't need these enzymes until I need these enzymes. The thing that's going to require those enzymes is actually going to flip the switch so those genes can be transcribed. That's inducible. What's the other one called? Repressible. Am I currently repressing it if it is repressible? No. No, I am not. How could I get it repressed if it's not repressed? That must mean I need to do something to the repressor, right? What could I do? Use a co-repressor. And that co-repressor then will shut it down. Usually that co-repressor is a product of an anabolic reaction. I have enough tryptophan, I don't need it anymore. Shut it off. Shut it off when you have enough of whatever that product is. Or turn it on when you need it. Those are your two options there. When you're dealing with what kind of control? Negative control. Okay? So when you look at this picture here, this is showing you an active repressor, and it's binding onto the operator, but it kind of spills onto the promoter. And so we looked at this one. Okay? So take a look at that picture. So in this case, there was an inactive repressor. RNA polymerase is on the promoter, and you're making the tryptophan. When you have enough of that tryptophan, what does it do? It goes and acts as a what? Co-repressor, which then activates the repressor and then shuts it off. Scenario number 2 we talked about, here it is. The Lac Operon, in this case, you have an active repressor on there. But when we need those genes transcribed, then who comes along? Lactose binds with it and then pulls it off so those genes can be transcribed and the lactose gets digested. What you have to be able to do on an AP exam is they will set up several scenarios and then ask you, is the operon on or is it off? So you have to look at all the scenarios that might turn an operon on or off. Another thing you want to then put on that extra layer, remember how we kept layering up, leveling up? Then we had to take into consideration the transcription factor needed in order to help RNA polymerase bind to the promoter. Even if lactose was there, it doesn't mean the genes are going to be transcribed, right? And so then we talked about you needed to have that transcription factor which would help RNA polymerase bind. What activates the transcription factor? Cyclic AMP. When do we have cyclic AMP? When
glucose levels are low.
Yep, so then it activates CAP, the transcription factor, so RNA polymerase binds. So those are all the scenarios that we need to have in play. Do you remember that one diagram where we had where it's, would weakly bind, or this is the only scenario where it would bind, when lactose is present and glucose is absent? Do you remember that part? Okay. Make it make sense to you so you don't have to sweat it out. If there are long statements about something, as soon as you get to a part in the statement that's wrong, dismiss that response for both me and for your AP exam. As soon as you read something, you go, that's wrong, stop reading. You can read it all. Obviously, read all your choices. But in your mind, go, okay, I don't have to worry about that one. I don't have to worry about that. Because this part of it is wrong. If any part of it's wrong, the whole thing's wrong, right? Don't go, oh, well, in this scenario, just if it's wrong, it's wrong. Levels of control. We then we transitioned into eukaryotic control. And eukaryotes aren't really dealing with operons. There's a lot of switches that we talked about. And the biggest part of all those levels of control is what? Which one is the biggest area of control? Yeah, transcriptional control. And in that scenario, it's not so much about negative control as it is about positive control and activators, and you have to have this present before that gene is transcribed. But we said in all these areas there are places where it exerts control. You could have failures in any one of those areas too, and you could have problems with a particular gene, but most of the time it works correctly. So we have, is the DNA even available? That was the one about chromatin structure, and we looked at things like what when we talked about that? Heterochromatin. Okay, which one is actively transcribed DNA? Euchromatin, right? Heterochromatin is tightly coiled and put away. What was our example of heterochromatin?
Bar bodies.
What?
Bar bodies.
Yes, bar bodies, exactly. Okay, and what are bar bodies?
The X.
Inactive X chromosomes. Do men generally, biological males, generally have a bar body?
No.
Who does?
Females.
Yeah, biological females have a bar body because we are a mosaic, because we have two X chromosomes, and in every one of our cells, one of the X's will condense. All right so far? All right, then what's the next level of control? Transcriptional control. And we looked at that already. We've talked about that you had to have several different, and that's the biggest one right there, about RNA polymerase getting to the promoter. What's the next level of control? Post-transcriptional. What are we dealing with in post-transcriptional?
The introns, exons.
Introns and exons. Because that's going to occur inside the nucleus. Inside the nucleus, right? Okay, what else? Cap and tail. What else? Leaving the nucleus. All of that is post-transcriptional. Yes, ma'am?
It talks about small RNAs and dsRNAs.
And almost three places it talks about it.
Yeah, so what I don't understand is where do small RNAs and dsRNAs take place in post-transcriptional control? Because I thought it says they bind to the ribosome. So what does that mean?
They can bind to the mRNA before it binds. So mostly translational control, or they can be byproducts of post-transcriptional control. Because when you cut out it could be right there. It could be a result of that.
Okay.
All right. We did that one. Translational control. So translational control is anything when you're dealing with ribosome and building your proteins. Exactly. How many ribosomal subunits do you have? Two. What one does it bind to first? The small ribosomal subunit and then the large ribosomal subunit. So anything that interferes with that. anything that interferes with the tRNA's ability to do their job and bring that amino acid. Post-translational control, what does that have to do with?
Three-dimensional folding.
Folding pattern. And who does the folding? Excellent. All right. Once we learned about DNA, and I'm not saying I've reviewed every single thing in that chapter. You're going to go back and look at that, right?
Yeah.
Once you've learned about the DNA and how it works, then we talked about modifying it, right? This is how can we use it for good or for evil. So we talked about some of the tools of a genetic engineer. So key tool of a genetic engineer is being able to cut DNA. What do you use to cut DNA? Restriction enzymes. Okay, so let's delve into that just a little bit. Restriction enzymes cut at a specific series of bases called a recognition sequence. Okay? So when they cut at that particular recognition sequences, you're assuming the more related organisms are, if you're going to do comparative biochemistry, right? The more similar their recognition sequence pattern would be, yes? And so you would expect them to have the same size fragments generated, which means that you would expect the same banding pattern in their what? Gel electrophoresis. Is there any part of that you didn't understand? Ask your bio buddy. Do you understand all the parts you just said?
Yeah, you need restriction enzymes.
I know you think.
Do you want to stick to it? You want me to? I don't know that I can do it exactly the same way. Do you need me to run through that one more time, or are you good? I think got it. One more time? Okay. So, you just fill in the blanks for me. Restriction, cut at a specific series of bases called a, and oftentimes they're called what where they cut? Starts with a P. Palindrome. Reads the same way forward and backwards, like race car. Remember that? Okay, so let's try again. Restriction, cut at a specific series of bases called the
Recognition sequence.
generating fragments of DNA. These the restriction enzymes that can be most valuable to us are those that create stickiness. Okay, because then later you can go and mix and match those, right, with the same restriction enzyme. If I am closely related to someone, then I have similar DNA, which means I have similar recognition sequences. Therefore, when you cut DNA, it should generate the same fragment length as a result, right? Because we're closely related, we have the same DNA. So therefore, when I take that cut up DNA and put it into a gel and turn on an electron current, the DNA sorts itself according to size. Good. Then when it sorts itself according to size, then what I can do is I can compare the banding pattern. I just read, maybe some of you already read this article. I read it, I think last weekend or last Friday or something. They are now taking fossils, and you should be able to understand this. It would be a great application level question on a final. They're taking fossils and isolating the proteins from fossils like collagen, and then they're cutting them with enzymes. One thing mentioned was trypsin, and trypsin and chymotrypsin come from your pancreas. They're digestive enzymes. They're endopeptidases. That's neither there. But they're cutting proteins. They're cutting proteins into fragments, right? Because proteins are made out of what? Amino acids. So they can cut protein sequences are going to be specific to each individual, right? Because who codes for those protein sequences? DNA. So therefore that DNA is coding for a specific series of amino acids. They're now snipping up proteins and doing gel electrophoresis with proteins. It's not exactly gel electrophoresis. What happens, they still send a current through, and so it dings at different times depending on the different size fragments. And from that, they can compare those two and go, oh, this is a fossil from here, this is a fossil from here, this is a fossil from here. Same idea, but doing it with proteins and very small pieces and being able to make comparisons. I just thought that was pretty interesting. Not using DNA, but using the proteins. Okay, same idea. Now, if you cut DNA at a specific series of bases, then you ought to be able to mix and match them, right? Mix and match them, and they should be what to each other? Complementary. But when you say cDNA, I'm really not talking, DNA is complementary to itself, but what is cDNA? What does that refer to when they talk about, oh, I made some cDNA?
Reverse transcriptase.
Yes, I'm talking about reverse transcriptase, right? Why might I need to do that? Why would I have to use reverse transcriptase? What scenario would I be in?
Bacteria can't process it.
When I'm trying to find what the DNA is that codes for a certain protein, perhaps, right? Or when I'm trying to insert a eukaryotic gene into a prokaryotic cell and they can't use the genetic junk, they don't know what to do, they can't cut it out because they don't have it, the same as us, right? The intergenic sequencing and spacing. Intra is within the gene, inter is between the genes. So you'll take your mRNA, your mature mRNA that's already left the nucleus, use reverse transcriptase to make complementary what? DNA. Yeah, and you can insert that into a plasmid and have a bacteria code for that protein. You work backwards from the mature RNA, so you cut out all the what? Get rid of all the what? Introns. So you only have the exons. Good. Okay. We talked about cloning, putting them into plasmids, and here was that part I jumped the gun. That what you have to worry about when you're using eukaryotic DNA is you have to have some regulatory regions, right, in order to turn the gene on. If you put it in there and you don't have it next to an operator, it's a silent gene. It will never get expressed. Yes?
Reverse transcriptase maybe is it doesn't have to be.
Right, that's one strategy. Okay, it must not have any introns in it because you can't express the introns. So this is where you might use reverse transcriptase. And then you need some way to get it into a cell, right? So really good genetic engineers are what? They're really, really small. They're not even considered alive. Viruses, yeah. Viruses, yeah. So that you can get it in, or you might use a plasmid to get it into a bacterial cell. Okay, questions or concerns on that? Okay, all right. We talked about cloning. We talked about other ways, some other things we can do to manipulate the situation. One is if I need lots of DNA, because I want to run some tests at a crime scene on a sample that I have, what would I need to do? Now before I don't have enough to do gel electrophoresis, so what do I need to do? PCR, right. PCR, to do PCR, polymerase chain reaction, polymerase, what am I thinking about when I think of polymerase? DNA polymerase, right? Polymerase chain reaction, I want to do several cycles using DNA polymerase. So what PCR does is it heats up to denature the DNA to get it into single-stranded, which normally we would use helicase, right? But now it's not in a controlled situation. We're doing it in a test tube. So if we just left helicase in there, it would just constantly separate them, right? So what you'll use is heat to denature it. And then what do you use? Use DNA polymerase. Where are we going to find DNA polymerase that can handle the heat? Thermus aquaticus, which is a bacteria that is archaean, that is used to that hot temperature. And we need some nucleotides in there and some primers to get the party started, right? And then we can just go through this heating where we denature, cool it to allow it to replicate, heat, cool, heat, cool, heat, cool, until we have multiple copies of the DNA. Then we can use some of our tools we talked about, using restriction enzymes to cut, using gel electrophoresis to separate, getting an RFLP, a restriction fragment length polymorphism, to get a whole bunch of, we can compare banding patterns from that. And we looked at this right here. Can you see this line and where the slash marks are? Yeah. Okay, so if you had this strand of DNA, and this is just the tail end of this is the tail end of the DNA. If I have 3 cutting places, how many fragments will I have if I have 3 cutting places? Yeah, because I have 1, 2, 3, 4 pieces of DNA. Do you see that? So whenever you take your section, like if you say, okay, I have this piece of DNA, and I cut it in 2 places, then really I have 1, 2, 3 sections, right? And that's what I would look for in my gel banding. Okay? And we talked about that. We looked at these different patterns. Just so we're clear on gel electrophoresis, the smaller sizes move the, okay, and the bigger pieces move the, so if I had two big pieces and one small piece, and one of the big pieces was slightly bigger than the other, could you think of a gel pattern that would make sense to that?
Yeah.
I have three pieces all together. One small piece is two big pieces. One of the big pieces is slightly bigger than the other.
Yeah.
So what would you expect?
The smallest piece to go the furthest.
Smallest piece out far, right? Then what about those two big pieces? Would they be close to that small one?
No.
Far away, and the bigger one would be the slowest, right? Then okay, good. See, you totally understand that. Perfect. We talked about gene therapy, ex vivo, in vivo. Anytime you're removing it, it's going to be what?
X.
Okay, and then we started talking about, here's chapter 18 or 15. 18? No, Miss Litton.
Not ready.
Not ready. We talked about change over time, right? We talked about evolution, and we talked about evidence for evolution. You remember all our pieces of evidence? Biogeographical evidence. Change over time, continents moved over time. You can only survive where you have an environment that you're adapted to. We looked at fossils, the tortoise, the turtle, Galapagos Islands. We looked at the shells. This guy must live on an area where he can eat algae from where? Oh, shoot it. From the ground, right? He's not eating tree leaves. How do you know he's not eating tree leaves? Yeah, because his shell would go up more if he was. We looked at natural selection. Natural selection, nature selects who's best fit. That assumes you have more offspring than you have resources for. Right? More offspring than can survive. And of those offspring, that they have variations amongst themselves that is in their DNA, that is heritable. And some of those variations make them better suited to their environment. What do we call those variations that make you better suited to your environment? Adaptations. Okay, and we are assuming that those who have the best adaptations are the ones that are going to survive and reproduce and have the best, what do we call it? Not heart rate, but it is your fitness, ability to pass on your genes. And we're assuming we have vast amounts of time. Yeah, vast amounts of time. Okay, in order to have those changes. Who changes the environment so fast that organisms don't have time to evolve? What?
Natural disasters.
People. Yeah, we change it so fast they can't even adapt. Okay? And we looked at all these pieces of evidence of evolution. You remember all of these?
Yeah.
Biogeographical, we looked at fossils. To be a fossil, you have to be over
10,000 years old.
Yeah, that was a piece of evidence. And we said in the fossil record, we can see gradual change over time, right? We can see transitional fossils. Did I have you learn all the eras in this one? I can't remember. Cenozoic, Mesozoic. Okay, that's different. Okay, never mind. Anatomical, there were three pieces of evidence.
Vestigial.
Okay, the first one I heard was vestigial. Vestigial is a structure that what? Found in ancestor, no longer functional currently. No longer functional. But it points to your ancestral past. Points to your past. It tells you where you may have come from phylogenetically. Okay, I heard another one you said homologous structures. I'm not looking at what structures? Not looking at analogous, but I am looking at homologous structures. Homologous structures have the same structure, same anatomy. I don't care what you do with it. I don't care if you swim or you fly with it. But if you have the same anatomy, if you're a homologous, homologous structure, same structure, then you probably have a common ancestor. Analogous just means we deal with our environment the same way. Spider has legs, I have legs. We both walk, but I'm not closely related to a spider. Yes?
Convergent evolution and analogous structures are basically the same thing, right?
Convergent evolution can explain analogous structures. Okay, what was the third one?
Developmental.
Yes, okay, and so that is, let's separate the two out because one is kind of biochemistry. I would say embryological, making embryological comparisons because we're talking about anatomy. So it is the development of embryos. But then when you talk about developmental evidence, usually you're talking about the genes and the Hox genes. We talked about how it's like the same types of genes for the different regions of the body, how we're segmented. and how we have those different genes and it's similar, showing common ancestry. But embryologically, we look like the more similar the embryos are, probably the more closely related in development. And also, it kind of looks like evolution on what? Fast forward, because we have
Gills.
Anna tail. Exactly. And then we compared genes, developmental genes. That was the other part. Biochemical is the one that's used the most now. If you can compare DNA, right? Similar DNA coding for similar proteins. We can even compare intergenic sequences, the non-coding sequences. And if our genetic junk is the same, things that you don't even select for phenotypically, right? Because natural selection can only select those traits that are expressed. Right? Then that would show some similarity. And then I just gave you an example of comparing proteins, right? The sequences of amino acids and proteins and how they use those to compare now as well. Okay, I think there's something else in that chapter.
Criticisms of
Yeah, I didn't ask you anything on this text. You're like, but I know those criticisms. All right, chapter 16. Chapter 16 was basically about Hardy-Weinberg. Let me see, what was the beginning of it?
Hardy-Weinberg.
Is it all Hardy-Weinberg all the time?
No.
Oh, let's go over our, yeah, that's still Hardy-Weinberg. Okay, can I please make sure you understand the big ticket of that? Hardy-Weinberg sets you up to fail. Sets you up to fail. Because it's saying you have to meet these 5 conditions if you want to maintain Hardy-Weinberg equilibrium. Since meeting those 5 conditions is so difficult, then probably you're not going to maintain Hardy-Weinberg equilibrium, which means you will evolve. Okay? Evolving within your species is called microevolution. Evolving to a new species is, and that's our chapter we're going to talk about tomorrow after your test, is macroevolution. I know you can hardly wait. What does it say on your calendar? How many times do I ask this? Now, Hardy-Weinberg says you have to meet these 5 conditions. Let's go back. 5 fingers of evolution, what causes change is having, what was the pinky? Small populations cause change, so if you want to avoid that, you need to have a large population. Okay? Do you understand the difference between that? Because since small populations cause change, 5 fingers of evolution, you need the opposite of that to keep it in equilibrium. Why is it that small populations cause change? Because they're more susceptible to what? Genetic drift, which is a factor of chance. There you go. There were 2 types. Don't say them out loud. Don't whisper them to your neighbor. See if your neighbor can think of them all by themselves. I randomly choose 6 of you out of this room to start a new colony in Oak Park Earthosphere above us. And those 6 I happen to pick out maybe don't represent the classroom. What is that called? Founder's effect. Okay? So that's when you have a new population started from a larger population where it doesn't necessarily represent the allele frequencies. What was the other one? Bottleneck. We're not, the ones that survive a crisis. Think about right here in Oak Park. There's all kinds of rodents that are up there. And we just had a fire, right? Only some of the rodents survived the fire. And those rodents that survived the fire are the ones that are going to repopulate these hills with their species. They may not be representative of the total population of rodents that were on the hillside. Yes?
So at the end of the day, it's the same effect on the population.
Exactly. End of the day, it's the same effect. End of the day, same effect. Yes?
Can you explain genetic drift? I didn't really understand.
Other than what I just now explained? Okay, so genetic drift has to do with chance. So I think of it like if you were on a boat and you're in England and you want to go to New York, do you want to drift there? No, purposeful, right? Purposeful direction, okay? Like directional selection, okay? But genetic drift is just you're drifting out there in the water. Okay, so that's what they call genetic drift. It's drifting due to chance, the chance of whatever current and the winds at that particular time. Smaller populations are more susceptible to drift than larger populations. The two forms of genetic drift are founder's effect and bottleneck. There you go. Okay? Small populations cause evolutionary change. Large populations protect you against allele frequency change. What's the next one? Non-random mating. Non-random mating says, I choose you. You have the traits. You're the biggest. You're the strongest. You have the best feathers. You dance the best. Okay? You put out the most blue things. You gurgle in the back of your throat or snap your claws in a way that is just so appealing to me. That is non-random mating. Random mating maintains Hardy-Weinberg equilibrium. Non-random mating disrupts Hardy-Weinberg equilibrium and causes evolutionary change. What's the third one? Mutations. So in order to avoid that, you need to have no mutations, or you need to have mutational equilibrium. I heard something. Did you say that? Awesome. Mutational equilibrium. Same amount of forward mutations equals the same amount of back mutations. Good. Just a second. No, I totally didn't. I have to get my steps after school. Okay. Mutation. What's this one? Gene flow. So, no, immigration or emigration. Which one is leaving? The Emma or the Emma? Emma. Think exit. That's how I remember it. Immigration, exit. Okay. What's the last one? Well, it's really adaptation, but adaptation has to do with natural selection. That nature is selecting who has the best adaptations. And then we had problems. Let's do a practice problem right now, for the heck of it. Here you go. Let's say that being right-handed is dominant over being left-handed. Being right-handed is dominant over being left-handed. In the United States, 90% of the population is right-handed. What I want to know is how many of those people of the United States are carriers for the left-handed allele. Okay, don't do it out loud, because then that takes away the thinking of other students in the room. Do you need me to repeat it, or do you got it? Okay. Let's say being right-handed is dominant over being left-handed. Currently in the United States, 90% of the people say that they are right-handed. What percentage of the people in the United States are right-handed but carriers for the left-handed allele? I'm pausing from. What did I just say? ¿Dónde está?
Where is it?
Okay, where is your brain? ¿Dónde está la brain? All right, here we go. Yo no sé. Tengo. Amas. Ignore me, I'm tired. Okay, come back to me. Is that what I really said? Yeah. Okay. First thing you want to do in these kind of problems, write down what you know. Okay, so I gave you this. 90% equals what? Because they will both show the dominant trait. P squareds would be like right-rights and right, and these would be right-lefts. Yeah? So I can't square root 0.9, right? I can't square root that because it has rights and lefts in it. So what do I know? What else do I, what can I, that's all I gave you. So what else could you deduce from that?
That 10% is the recessive.
Do you agree with that?
Yes.
How do I write 10 as a decimal?
0.1.
0.1, okay? Equals my what in my equation?
Q squared.
So great. Now what do I do?
You square root it.
Square root it. And now, sorry, I have to write it like this. Q equals what? 0.32. And I know that P plus Q equals 1, because if I reach in that gene pool and 32% I get a Q, then what do I have for P?
0.68.
What is it?
0.68.
Okay, now I asked you what percentage of the population is a carrier for the left-handed allele. So those are my 2 PQs. So I just say 2 times 0.68 times 0.32 will equal my 2 PQ. And what is that?
52%.
Yeah, it was 43.5% is what I got. 43.5. Yes.
So if
I can't hear. Shh, go.
So if it's
Shoot, did I start it?
type where it's like codominant, how would you be able to use this? Because in blood types A and
That's different. So you have to look, and also that's an example where you have multiple alleles. Here I'm assuming there's only P and Q. So you don't have to worry about it for anything else. Yeah. Okay, what else? All right. So then this takes us where you want to, and we don't have time to go over all of them, but I still want to address them with you. Because you're not having an essay. Can I still have you? You're not having an essay tomorrow, but you are going to have an essay by the end of the week, right? I got to write that final. Okay. Right? Oh, is that next week? Oh, oh yeah. Miss Liddon. By the end of the week. Okay, wait. Let's just finish this. Because there are several associated with this one. If you look, remember I've given you the 2 essays, right? The new school and old school. Both of them have shown up on your test. So one in here, oh no, not that. There's one in here about making comparisons. I recommend that you do this. You don't have to take a picture of this. I gave you this at the start of the school year. Remember? Start of the school year for review. So you have an essay right here about who's more closely related. You have 3 questions about restriction enzymes or structure of DNA, about carbon dating. Remember half-lives? I know because I taught it to you last year. Half-lives? The time it takes for half an isotope to decay? Okay. And then there's one in here about, oh, you know how to do these. Look, population, populate, right? These are Hardy-Weinberg ones. This is all in the new school ones. And describe 3 types of evidence that scientists use to discover evolutionary relationships between organisms. This is the old school essays that you could be practicing for. Now, this should not be the first time you're looking at these, right? What have I said to you? Do it when? As you go along in the material, right? Because that helps you prepare for your quizzes and your tests. So there's things here about what is an operon, about gene expression. Here's things about restriction enzymes. Here's another question about restriction enzymes. And then here's a PQ one, Hardy-Weinberg, and then here's modern theory of evolution. So all of these are here. Those are all game, right? And we need to keep in mind those as well as unit 3, right? So, and we already went over those, but I'm just reminding you, those are all the ones that you have to look at. Plus any related to this last chapter. Okay, but what kind of test are you taking tomorrow? Multiple choice. How do you prepare for multiple choice? Yeah, end of the chapter, right? And also make sure you can do Hardy-Weinberg's. Yes?
What about the Hardy-Weinberg lab? Looking it over.
You look it over so you're familiar with what we did, but it was just, you need to just be familiar with what we did. Okay. Which was just more Hardy-Weinberg scenarios. Yeah.
Thank you.
Okay, don't stay up too late. Have a piece of toast.
Potential problems tomorrow on Hardy-Weinberg.
Yes, you could do your highly suggested reading and thinking. Highly suggested reading and thinking as well.